Okay, so I'll start my session now. So you can visit this is visible, right? Okay, fine. So this is our topic today. So here what we are going to do is, as you already know, that we are having some objective of the study. So today, what is the objective? The objective is to identify the reason behind the genetic disease. Okay. So we have identified a particular disease which is very rare. Okay. So what we'll do is we'll talk about those things as well. Okay. Just hold on. Okay. So what happens? Just hold on. Okay. So we'll be talking about a thing called, listen very carefully, metabolic diseases, or shall we say inborn errors of metabolism. Inborn inborn errors of metabolism. Okay. So as the name suggests, these are diseases where metabolic abnormalities are there. Okay. And since these are inborn errors, therefore these are nothing but genetic diseases. Okay. Genetic diseases and cannot be cured. Okay. Genetic diseases, okay, and is not very likely to get cured. Okay. Now these are genetic diseases, but these are not exactly the same like thalassemia and all other things. Okay, sickle cell anemia, thalassemia, these are different kind of diseases. These are also genetic diseases, but not inborn error of metabolism. Some common examples of inborn error of metabolism are I think you already know, for example, alkaptonuria. Okay, alkaptonuria. Okay, in this disease, what happens? From urine, a derivative of tyrosine metabolism is produced. So it's a abnormality of tyrosine metabolism. Okay, tyrosine metabolism abnormality. Okay, abnormality. So this is a classical case of what we call is genetic disease, which is error of metabolism. Okay, but there are some diseases. This is comparatively not very rare, but there are some diseases within these class. What are the class? The class is this. These are inborn error of metabolism. These are genetic diseases. But there is one more class which is called, okay, uh, orphan diseases. Okay, orphan diseases. Why orphan disease? Because these are very rare inborn error of metabolism. Okay, these are rare diseases. So one example of such rare disease is our case study today. Okay, listen carefully. So you may ask, sir, why we are discussing this? Because there are many rare diseases. If you choose one and if you start to work on that, there is huge potential that you can end up with really some very good insight or very good research you can do. Okay. So this one of the example of this orphan disease is called Barth syndrome. Okay. So Barth syndrome is a rare genetic disease. Okay. Now what is the manifestation? Manifestation means what are the signs, symptoms, and how these are diagnosed. So the sign and symptoms are cardiac abnormality. Okay. Cardiac abnormality in young, what do you call, young individuals. Okay, or shall we say in very small age. Okay, abnormality in young age. Yeah, young age. Okay, very young age. So the patient usually dies before the patient usually dies before 10 years, okay? Usually before 10 years, the patient will surely die, okay? There is a very rare case that where the patient has survived. Now, what is this thing behind this? What is the reason behind this? The reason behind this is a mutation. The reason behind this is a mutation in cardiolipin metabolism. Cardiolipin metabolism. Okay. Now, cardiolipin is a lipid which is present in heart. Okay. This lipid is present where? In heart muscles or cardiocytes. Okay. Cardiomyocytes. Okay. In cardiomyocytes, these particular lipid is present. So due to the abnormality of the production of this cardiolipin, what happens is this genetic disease occurs. Okay. So on 2012-13, we undertook this particular study where our objective is to identify why this disease takes place and is there any what you call supportive therapy that can be identified. Okay. So see the topic. The topic is missense mutations in tafazin that escort to mild clinical symptoms of Barth syndrome is owed to the minimal inhibitory effect of the mutations on the enzyme function in silico evidence. So what happens? We identified that there are variety of the exposure of the disease. Some people are there who will instantly die. Okay. Some people are there who will survive for a little bit, but eventually they may also die. Okay. So what was the reason behind that? We found that there is a protein called tafazin which is very important for cardiolipin metabolism. So that protein is mutated, and if the mutation is limited means if the mutation do not alter the structure of the protein rapidly, then what would happen is there will be some mild clinical symptom. Okay. So mild clinical symptom means the patient will not complain for severe cardiac abnormality, and the patient will somehow will have not very alarming what we call situation. Okay. So this was the study that we did in 2015. Okay. So it was been published by Springer. It's a very important and very what we call well-known publisher of science, biomedicine, biotechnology, medical field, and this study has been cited also in multiple. As you can see, in this particular this thing only, there are 5 citations, and there has been many access also. So many people have tried to study from this. So in Web of Science there are 5 citations, and in cross reference there are 2 more citations. Citation means for example, you have identified a particular problem, you have published a research paper on it. So if somebody comes back to your research and searches for the information that you have gathered, indicates that your research has some value. Okay. If the citations are not there, it means that it could be a problem, there could be a problem, but the problem is not very important. Okay. If the citation is very, very high, it means that it's a very major problem. So as I have said that this Barth syndrome is a disease which is very rare. Okay, rare genetic disease. So as we have named also that orphan disease. Okay, it's an orphan disease. So in these cases, 5 citations indicates that throughout the world, 5 different areas or 5 different universities have used our study for their further course of action. Okay. So in this what happened, we identified that the protein which is called as tafazin is a phospholipid transacylase. So phospholipid transacylase is a class of enzyme. Okay, it's an enzyme, and it is present in the TAZ gene, TAZ gene. Okay, and the cytogenetic location is where the Q arm of the 28th chromosome. So as you can see here, cytogenetic location means in which chromosome the gene is present. Okay, the gene is present in, okay, all the chromosomes are having two legs. Okay, so this is basic also, but still I'll draw. If you can remember, tell me if you can remember this structure. Okay, so chromosomes are look like this. Okay, chromosomes, no, these are like this. Okay, so there will be a centromere in between. This is the centromere. Consider that this is the centromere. Okay, so there is a p arm in all chromosomes. There is a p arm and there is a q arm. Okay, and q arm. So our gene of interest is present on the q arm. Okay, so our gene of interest is present where? Is present in the q arm. Okay, somewhere here. Just hold on. Okay, so we'll identify this as our this thing in this q arm. It is present. So now what else? The mutation of this protein causes a disease called Birth syndrome. Okay, so Birth syndrome is also having a complete name. See the name: 3-methyl glutathione aciduria type 2. Okay, so Birth syndrome is the name which has been proposed by a scientist called Barth. Okay, but classically the name of the disease is 3-methyl glutathione aciduria type 2. That means the patient will release a metabolite which is methyl glutathione or 3-methyl glutathione compound, which is released in the urine of this patient. Okay, see here, the inborn error of metabolism often lead to fetal or infantile fatality. So what does it mean? It means that in the fetus only the patient will die. Consider that this is the, okay, just hold on. Okay, this is the mother. Okay, so what happens during pregnancy? The fetus develops inside. Okay, and with the mother's body and the what we call is the fetus body, there is a connection between the mother's body and the fetus body. Okay, which we call as let us call it uh, 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 the the fetal connection. Okay, the fetal connection with the mother. Okay, so what happens uh, uh, due to the mutation? Due to the mutation, what would happen is all the things will be okay, but there will be major heart problem in the fetus. Okay, and within 3 weeks of pregnancy, the fetus will die. Okay, or if at all the fetus takes birth, it will die within the infant age. Infant age means before 6 months, before this thing 6 years, the patient will usually die. But in some cases, here you see, some cases it leads to mild clinical symptoms. Okay, so it leads to what? It leads to mild clinical symptoms. So mild clinical symptom means the patient will eventually die, but it may survive up to 10 years. Okay, I have not mentioned. Yeah, it may survive up to 10 years. Okay, so now we'll see how the protein looks like. Okay, so coming back, I'm sharing this link with you. Okay, in the chat box, you also will click. So this is the sequence of tafazine protein. Okay, so tafazine in human beings is a transacylase, as you can see here. Okay, so these are different tafazines present. So we will take for Homo sapiens. That means human tafazine we will identify. So this is human sequence, full protein. Okay, this is partial protein. This is also partial. So we will take the full protein. Okay, so this is the full protein which is having approximately 262 amino acids. Okay, Let us hope that the page opens up. Many a times what happens, it do not respond. Okay, it will open up. So what we are trying to do, we are trying to figure out the structure of tafazine, okay, human tafazine, and we will try to figure out the active site of tafazine where the reaction takes place. Okay, so here there is a mentioned, see, crystal structure of tafazine is not yet resolved experimentally. Okay, so in this database it is highly unlikely that you will get the sequence. Okay, so it is highly unlikely that you will get the exact sequence. Here you see, when we searched with tafazine, we searched this tafazine in RCSB PDB, we found a structure, okay? But the structure, no, it is completely different. Here you see, there may be some similarity, but it is not exactly the one we wanted. So here you see, it is the name of the protein. Early PP module assembly intermediate of complex 1. Okay, that also of a different organism, Yeroia lipolytica. So we don't want this structure. So what we want the exact structure of tafazine to be built. Okay, so let us see whether this there is some problem with the database itself. So what we'll do is we'll model this tafazine and then we'll do our study. Okay, so we are recreating the study that was been published in 2015. Okay, likewise you can also identify some genetic diseases and then eventually you can start your research. Now here you see you are having the structure of the protein which is having 262 amino acids. Okay, so it is human. Here you can see TAS human, that means tafazine human. It was been published very recently. See here, it has been updated in May 29, 2024. Okay, so it has also been published, I think. Yeah, it is also been published. Nature Genetics. Here you see, the study has been published in Nature Genetics. Nature is a very big this thing, research journal. If something is appearing on Nature, it is a very impactful research. Okay, so here you see the research name, title: Novel X-linked gene G4.5 is responsible for birth syndrome. Okay, so this is a very important study. They might have used our study also. Sometimes some scientists will cite it. That means they will give credit to us that we have already done the study. Not exactly the same, but some study we have done. Afterwards, whatever is left, they have done this because they are having more resources. We have no, whatever resources we had, we published. Okay, so they may have or may not have cited our work, but no problem. They have already worked it, and then eventually they have deposited the sequence. So what we'll do is we'll click on FASTA and we'll have our sequence. Now in this sequence, as you can see, these are all single amino acid, what do you call, letters. Okay, can you tell me what is the first letter and what does it indicate? Yesterday also I have spoken. Who can tell me? Can anybody tell me what is this first amino acid? The first amino acid is written as M. So it indicates that this is the amino acid methionine. So methionine is also the initiation codon, and therefore the protein is complete. It started with an M and ended somewhere. That means its N-terminal residue is okay. So here what we are discussing this study which we have published in 2014-15. It is been published in Springer. It's a very good journal. The citation is, the impact factor is approximately 3.824, and the journal name is this: Interdisciplinary Sciences, Computational Life Sciences. Here what we have done, we have identified the protein tafazine. We have, because the crystal structure is not there, we modeled it and eventually identified the mutation which leads to what you call minimal inhibitory effect of the or on this particular protein. Okay, it's a rare genetic disease and it is also called as inborn error of metabolism. The patient eventually die in very young age, but some patients has been seen to survive up to 10 years. So we have studied those particular mutations. Okay, okay, if there is any disease doubt, you can ask me.